The year is 1878. Northern Italy. Chickens on a small farm begin to die, their bodies racked by a mysterious illness. Farmers, at a loss, watch their flocks dwindle, the casualties mounting each day. What they are witnessing is the first recorded outbreak of avian influenza, a disease that will haunt humankind for over a century. This is not just a story about sick birds. It is a story about a virus, one with the chilling potential to leap from animals to humans, unleashing pandemics that could cripple our world. It is a story of science and ignorance, of global interconnectedness and the fragility of our ecosystems. Avian influenza, often called bird flu, is a stark reminder that the health of animals and humans are inextricably linked. What begins as a whisper in a chicken coop can quickly escalate into a global health crisis, demanding our attention, our resources, and our unwavering commitment to prevention and preparedness. The story of avian influenza is one of constant evolution, a relentless dance between a virus and its hosts, with humanity caught in the crossfire. Understanding this complex interplay is not just an academic pursuit, it is our best defense against the next pandemic. Avian influenza is not a single entity, but a group of viruses, classified as type A influenza viruses. These viruses primarily infect birds but can also infect other species including humans, under certain conditions. These viruses are masters of disguise, constantly evolving into new strains with varying levels of virulence. This ability to change makes them particularly challenging to control and predict. Some strains cause mild or no symptoms in birds, while others, like the highly pathogenic H5N1, can wipe out entire flocks within days. The impact on poultry farming can be devastating, leading to significant economic losses. These viruses are categorized by two proteins on their surface, hemagglutinin, H, and neuraminidase, N. These proteins play crucial roles in the virus's ability to infect and spread. There are 16 known H subtypes and 9 known N subtypes, leading to a dizzying array of potential combinations, each with its own unique characteristics. This diversity makes it difficult to create a one-size-fits-all vaccine. The H protein acts like a key, allowing the virus to attach to and enter host cells. Without this key, the virus would be unable to initiate infection. The N protein, on the other hand, acts like a crowbar, helping newly formed virus particles escape from infected cells to spread the infection. This process is essential for the virus to propagate and infect new hosts. The ability of avian influenza viruses to mutate and swap genes with other influenza viruses makes them a constant threat. This genetic flexibility allows them to adapt to new environments and hosts. This process, known as reassortment, can lead to the emergence of novel strains with pandemic potential, capable of infecting humans and spreading efficiently between people. Such events are rare, but can have catastrophic consequences. Understanding the genetic makeup and behavior of these viruses is crucial for developing effective vaccines, antiviral drugs, and control measures. Ongoing research is vital to stay ahead of these ever-changing pathogens. It is a race against time as each new mutation brings us closer to a potential pandemic. The scientific community must remain vigilant and proactive to prevent such a crisis. The 1878 outbreak in Italy marked the beginning of our understanding of avian influenza. This was a time when the world was just starting to grasp the complexities of viral diseases and the impact they could have on both animal and human populations. Since then, the world has witnessed a series of outbreaks, each one a stark reminder of the virus's ability to adapt and spread. These outbreaks have not only affected poultry but have also had significant economic and social impacts, disrupting livelihoods and causing widespread fear. In 1959, a new subtype, H9N2, emerged in chickens in Hong Kong, highlighting the virus's ability to jump between species. This event underscored the importance of monitoring animal health as a key component of preventing human disease. Then, in 1997, the highly pathogenic H5N1 virus crossed the species barrier in Hong Kong, infecting 18 people and causing 6 deaths. This was a wake-up call for the global health community, illustrating the potential for avian viruses to cause severe human illness. This outbreak sent shockwaves through the global health community, raising the specter of a devastating human pandemic. It led to increased surveillance and research efforts to better understand and combat these viruses. The H5N1 virus continues to circulate in birds and occasionally spills over into humans, with sporadic cases reported in Asia, Africa, and the Middle East. Each new case is a reminder of the ongoing threat posed by this virus. While human-to-human transmission of H5N1 remains limited, the virus's high mortality rate, around 60% in confirmed cases, makes it a serious public health concern. This high fatality rate underscores the need for continued vigilance and preparedness. Over the years, other subtypes such as H7N9 and H5N6 have also emerged, causing human infections and raising global alarm. These subtypes have shown the virus's ability to evolve and present new challenges to public health. Each outbreak serves as a grim reminder that avian influenza is not a problem of the past, but an ongoing threat that demands our constant vigilance. The virus's ability to mutate and spread means that we must remain ever watchful and prepared. The history of avian influenza is a story of emerging threats and evolving responses. It is a testament to the resilience and adaptability of both the virus and the global health community's efforts to combat it. It underscores the need for robust surveillance systems, rapid response mechanisms, and international collaboration to detect and contain outbreaks before they escalate into global crises. Only through coordinated global efforts can we hope to stay ahead of this ever-evolving threat. The avian influenza virus is a master of adaptation, constantly changing to exploit new hosts and environments. This relentless adaptability is what makes it such a formidable pathogen, capable of crossing species barriers and spreading across the globe. This adaptability is fueled by two key processes: mutation and reassortment. These mechanisms allow the virus to evolve rapidly, outpacing the immune responses of its hosts, and sometimes even overcoming antiviral treatments. Mutation is a constant process in all viruses, with small errors creeping into the genetic code during replication. These errors, or mutations, occur randomly and can accumulate over time, leading to significant genetic changes. Most mutations are harmless or even detrimental to the virus. They might result in a less effective virus that is unable to replicate efficiently or spread to new hosts. However, occasionally a mutation arises that gives the virus a survival advantage, such as increased transmissibility or the ability to infect new host species. These advantageous mutations can lead to more virulent strains that pose greater threats to animal and human health. Reassortment, on the other hand, is a more dramatic process that occurs when two different influenza viruses infect the same cell. This can happen, for example, in a pig that is co-infected with both avian and human influenza viruses. During this process, the viruses can exchange genetic material, creating a hybrid virus with a mosaic of genes from its two parent viruses. This new virus can have unique properties, potentially making it more infectious or able to evade the immune system. This reassortment process can lead to the emergence of novel influenza viruses with pandemic potential. These new strains can spread rapidly through populations that have little to no immunity against them. For example, the 2009 H1N1 pandemic virus, which caused the first flu pandemic of the 21st century, was the result of a reassortment event involving genes from swine, avian, and human influenza viruses. This highlights the unpredictable nature of influenza virus evolution. Wild birds, particularly waterfowl like ducks and geese, play a crucial role in the spread of avian influenza. These birds are natural reservoirs of the virus, often carrying it without showing any symptoms. These birds often carry the virus without showing any symptoms, acting as silent reservoirs and spreading the virus along their migratory routes, which crisscross continents. Their long-distance flights can transport the virus to new regions, facilitating its spread. This avian Autobahn connects distant corners of the world, facilitating the global spread of avian influenza viruses and increasing the risk of spillover events into poultry and humans. Understanding these pathways is crucial for predicting and preventing future outbreaks.